Welcome to the fourth phase of water with Gerald Pollack. I'm Doug Cook, NOFA Mass Education Events Coordinator. I'll be your host for today. We are going to get started by using this screen, which I'm sure you're familiar with as we've used it for many of our events, to help us remember that we are occupying land that has been historically colonized by indigenous people for many thousands of years before European colonization. And we encourage you to use this link to learn more about the indigenous people in your area of what is now known as America, and follow those links to learn more about their continuing structures and struggles that you may be able to support. As well, we are reminding everyone that you are entering on mute. Please remain on mute during the presentation. We may invite you at some point to come off of mute and just use that little microphone button to ask your question. And then once you're finished, go ahead and remute yourself to preserve sound quality for the recording. As this is being recorded, it will be also available later on our YouTube channel. We will have you entering questions into the chat box throughout the presentation. I'll be monitoring those questions and I'll bring them back at the end for the question and answer period. We like to remind everyone to thank our sponsors. We are very grateful to their support. They're doing some excellent work and have excellent products. So the next time you see someone or you're giving feedback to one of these organizations, please remember to say how much you love their support of NOFA Mass. We hope that you are enjoying this event as well as the online auction. There are some really cool items that you can bid on, so go ahead and check those out in between sessions. As well, we encourage you to spend some time in the virtual vendor room, learning more about many of our vendors and their amazing work and products. So thanks again for all of our vendors for being here. And with that, it's my great pleasure to introduce Gerald Pollack. Jared, he received his PhD in biomedical engineering from the University of Pennsylvania in 1968. He then joined the University of Washington faculty and is now professor of bioengineering. He is also a founding editor-in-chief of the journal Water, convener of the annual conference on the physics, chemistry, and biology of water, and executive director of the Institute for Venture Science. Thanks for joining us, Jerry.
Oh, it's my pleasure, Doug. It's really nice to be here with you. And is it time for me to share my screen?
Yeah, when you are.
Yeah, here we go. Let me just move this up here. Okay, so I would like to talk to you today about the fourth phase of water, and that water's central role in health and also in agriculture. And one of the surprising things that I'm going to talk about, perhaps it's not surprising for some of you, is a surprising role of light. The idea of light is going to be woven into the fabric of what I will present. So, whoops, why am I not advancing? Okay, this is the way. So the topic is water, and you know, when you think about water, I think most of us presume that whatever there is to know about water must already be known. After all, how could there possibly be questions that arise? Scientists have been studying water for several hundreds of years. So we must know all there is to know about water. And so I first addressed that, and that do we really know everything there is to know about water? And let me give you a few examples of what, if we really knew all that there is to know about water, the questions that I'm going to pose to you should be directly answerable by any of you. And here's a cloud that's sitting above above the water. I know the weather where most of you are right now is lovely. Probably looks just like this. And there's this little cloud sitting here. Now, a question that maybe you've never thought of, or maybe you have, is, well, the cloud is made of water. Water is heavier than air. If I were to climb up a ladder all the way up here, take a pail of water, turn it upside down, the water would come pouring down like a waterfall, but the cloud floats. It's just water, basically, and the question is, well, why do clouds float? And if you don't know the answer, then, well, probably you don't know all there is to know about water. Now, here's another example. These are two beakers, lip to lip. Both are filled with water. You stick one electrode here into one of these beakers, another electrode here, and you impose a high voltage, and this is what happens. So a bridge forms between the two, and if you move one beaker away from the other, just pull it away using a moving stage, you can see that this bridge persists. And it persists indefinitely in general, and it persists for distances or spans as much as 3 centimeters, sometimes even 4 centimeters. How on earth is this possible? Water, as we know it, doesn't behave this way. Can you explain it? And if you can't explain it, then you really don't know all there is to know about water, because this is easily reproducible. I'm sorry, okay, it's not acting in the usual way. Then we have another example. This is a lizard that lives in Central America. I think it's Costa Rica. And it spends a good deal of its time sitting on tree branches. When it gets inspired, and I have no clue what inspires it, it will jump from the branch, and it will jump basically into the water, and it will walk on water. And that's why it's colloquially known as the Jesus Christ lizard, walks on water. How can it walk on water? And if you can't explain it, then you don't know all there is to know about water. Another one is one of my favorites. This was a demonstration that I saw in Tokyo, courtesy of Professor Shogo Ueno. And Professor Ueno, Tokyo University, took a container, a trough of water, and he squirted some red dye into it for reasons that will become obvious in a moment. And behind it, he has a superconducting magnet. And he turns on the magnet, and the Red Sea splits, as you can see here, so that Moses could easily walk across that Red Sea. This is a real demonstration. This is not Photoshop or anything like that. And, so the question is, what's going on here? And the subtext is, well, gee, if you can't explain this, you don't know everything there is to know about water. So I hope I've made my point. We started our interest in water based on inspiration from a guy named Gilbert Ling, who passed a couple of years ago, just shy of 100 years old. And Gilbert came from China. He was in the first cohort of Chinese young students chosen from throughout China. They chose 3 to come to the US to study from all of China. And there was a chemist, a physicist, and a biologist, and Ling was the biologist. The physicist went on to win a Nobel Prize in physics, and they all thought that Gilbert Ling was probably the cleverest of all 3 of them. So this is a rather formidable intellect guy, and he spent a good deal of his academic life studying water, and he came to the conclusion from many experiments and theoretical work, and having written many books, he came to the conclusion that in biology, especially inside the cell, that the water was not like water in the glass that I'm holding, which I guess you can't see because, well, whatever. where the molecules are bouncing around, a gazillion times per second, and they're randomly oriented and such. It's not like that at all. He said inside the cell, the water molecules are lined up like soldiers at attention. And obviously this was a radical point of view, because the prevailing point of view is that the water in the cell is like any kind of liquid water that you drink. And I happened to meet Gilbert at a conference. I was invited to a conference in Hungary. I had known about him in the past. I'd heard, but finally meeting him and listening to his presentation really impressed me. I was impressed by the logic of his approach. I was impressed by the evidence that he presented, and even more than that, I was impressed by the dozen or so people who had evidence in support of Gilbert's ideas. And so I was moved when I met him, and I thought, my goodness, if this guy is right about biological water, if he's right, this changes everything. It's so fundamental, because if you assume that the cell consists of liquid water, then your line of thinking will proceed along in one direction. But if you think it's not liquid water, but a different kind, almost like a crystalline water, then everything changes. And so I'm thinking, what can I do? I had been studying the molecular mechanism of muscle contraction, and I, of course, was enchanted by so much interesting stuff in that field, but water seemed more interesting. So the first thing I did when I got back home to Seattle from that conference in Hungary is to give one of his books to some of my students, the ones who could understand. And mind you, his books were not so simple. They're really formidable to understand, but somehow my students were eager to read, and they came back to me, and they all had the same impression. This is really important stuff. I don't know if he's right, but looks like, based on the evidence, he's got something really important to say. And if he's right, it's going to be amazing. So I couldn't stop. I was so intrigued by all that. The first thing I thought I would do is try to make clear, or clearer, Gilbert Ling's ideas, because Gilbert Ling, bless his heart, when he would write a book, he wrote about 7 of them, I think. He'd sit down at the typewriter at first, and eventually the word processor, and he'd bat something out. And I kind of think he never submitted his stuff to an editor, or never went through his own stuff thinking that, by gosh, everybody is equally brilliant as he is, and therefore there's no reason to edit. And so the end result is a book that takes a lot of attention to go through and understand fully. So I set myself on a goal to try to make his ideas understandable to the world. And I wrote this book. It was way back in 2001, Cells, Gels, and the Engines of Life. And the book got mixed reviews. On the one hand, it did win a prize, but on the one hand, some of the critiques opined that, oh, this is the same old Gilbert Ling stuff. And we know Gilbert Ling is a crackpot. He has this weird idea about water, and therefore don't bother reading it. Those were the negative reviews. The reviews were rather polarized. And on the positive side was a review by someone from Harvard University, therefore he must be right if he's from Harvard. He wrote something like, this is a 305-page preface to the future of cell biology. That one I liked. So anyway, mixed reviews, but having dealt in some detail with the ideas of Gilbert Ling, and the book went actually somewhat beyond Gilbert Ling, suggesting that this kind of structured water, as he called it, was central for everything the cell does. It played an integral role in the function of each and every cell. So having written the book and having professed so radical, you might say, radical an idea, I felt it was time to start doing some experiments. And so essentially, I changed fields 2 decades ago. And the book, by the way, for discerning people, you know, they will pick out this book among other possible choices. And this one, this guy is still reading. I guess he's enjoying himself doing it. Not my child, but child of a student of mine. So wishing to pursue the ideas of Gilbert Ling, and I will tell you at the outset that we finally came upon evidence that while Gilbert was on the right track, there's some things that are fundamentally different, and I'm going to be telling you about those. But anyway, according to Gilbert Ling's central idea, inside the cell, you have solids like proteins, and on their surfaces are charges that are distributed throughout the surface, making them generally hydrophilic or water loving. And then you got water molecules, and each water molecule is represented as a dipole. So you have a dipole here with plus, light colored, and minus, and another one here, the same. And you could see how the first dipole would line up with this negative charge. And because of the first dipole, that would tend to align the second dipole and the third and so on. And so you can envision a kind of ordering of these dipolar water molecules. But eventually, when you go far enough, because of Brownian motion, thermal motion, that is the motion that's inherent to all molecules, you expect that the order will be lost. Physical chemists will, in general, mainstream physical chemists will admit to perhaps one or two molecular layers of ordering, but beyond that, nothing. And Gilbert Ling said, no, you guys are wrong. And he professed that the ordering can extend out to many, many molecular layers, perhaps even hundreds of molecular layers. And that would be entirely different. So for us, in thinking about doing experiments on this, we were thinking about this model, looking for order. And if there's order, like a crystal where the atoms or molecules are lined up, it tends to exclude solutes and particles. You know, when ice forms, you get a pure ice crystal. And in order to get pure ice crystal, it has to get rid of all of the contaminants. Otherwise, you have contaminated ice, not pure ice. And so this is characteristic. So we were looking for some region in a preparation where particles or solutes would be excluded. And we found it. And we found it rather quickly, thanks to a few lucky quirks on our part. What we did was we put a gel, which you see here, into a container. And we added to the container water next to the gel, water plus particles. And these particles were little spheres, and they're called microspheres because they're micron in size. And so we started with a gel next to a suspension, an aqueous suspension of microspheres. And we looked through the microscope, and we saw immediately that there was a region here that contained none of these particles. And we thought, well, it's possible that this is a region where the water molecules, this is water after all, where the water molecules are lined up, as Gilbert Ling had suggested, and therefore they pushed out these contaminants or particles. And this was 10 micrometers, which is sizable by molecular standards. And we had no evidence for it, but we thought, well, perhaps this is the case. Perhaps we've identified what Gilbert Ling was talking about. And the situation was actually more dramatic than that. If you look at the video, sped up, of course, you could see that something is growing here. This is a period of about 5 minutes, growing and pushing these spheres out. And finally, the spheres would settle in at a distance on the order of 50 micrometers. And we saw this again and again, and we shared it with many people. And an Australian colleague opined that we should give it a name because you can't go through this elaborate description. It's easier to call it something. And he suggested calling it exclusion zone because it is a zone, it's a zone of water that excludes, it excludes particles. Later we found it excludes virtually everything. And the water here we called EZ water. And EZ works really well because it's easy to remember. The guy who was invented in Australia, I think in Australia it's EZ, not EZ. Certainly is in a lot of countries, so it doesn't work as well. But anyway, we sometimes refer to this as EZ water, and later I'll mention that we call it also fourth phase water because the water here is really different from ordinary water in a glass. So in terms of generality of this finding, we investigated many hydrophilic or water-loving surfaces, and actually most hydrophilic means water-loving means if you just have a horizontal surface and you drop water on it, the water will spread out, as opposed to hydrophilic, hydrophobic, water-fearing, like Teflon. You drop water, it beads up. So this works only with hydrophilic surfaces, which includes most surfaces to some degree. And we found that many solutes are excluded. So many surfaces create the zone, many solutes are excluded. It's very general. It even occurs in xylem. So this was sent to me many years ago, this electron micrograph. And sorry, I can't recall the species, but you can see this is 5 micrometers. And this is a xylem channel or tube into which Martin Canny, who was perhaps the premier researcher in terms of vascular flow in plants, he sent me this and he said, yeah, there's definitely an exclusion zone here in the xylem because you can see that the particles are confined to the center. At the edges, you see very few, if any, particles on both edges. So it looks as though whatever this exclusion zone means or EZ means, it exists in xylem. And so this is, for me, an interesting, shall we say, finding. So is this, next question though is this water that we're talking about, this EZ or fourth phase water, is it really different from bulk water? I've alluded to the possibility that indeed it might be different from bulk water, but I presented no evidence. So here's a list of evidence, and evidence that EZ water is physically different from bulk water. And I'm not going to go through all of this because it's published and also it's old. This is the list that was compiled about 10 years ago. And by now, the list would be double this size, but if I were to take you through all of this, we'd use up all the time. So I'm not going to, but just to give you some idea, for example, we found that EZ water molecules are more constrained than ordinary water molecules, and we were able to find out using nuclear magnetic resonance, and so on and so on. This is an important one. It absorbs light at 270 nanometers, whereas ordinary water doesn't do that. It's more viscous, et cetera, et cetera. But I just want to refer to this one because this is absolutely central to everything that I want to say, that the EZ is not neutral like ordinary water. It has negative charge. And let me explain. We found this by some coincidence. We're using the same electrodes that were invented by Gilbert Ling, the same Gilbert Ling for which he should have won a Nobel Prize. And we had a situation that looked like this. So you have a hydrophilic surface sitting next to water, and the exclusion zone is right here, and here's the water beyond the exclusion zone. See, initially this was just water, and during those 5 minutes or so that I showed you, this is what happens. And we stuck these tiny electrodes that come to a point less than 1 micrometer into various regions, and by so doing, we found that typically this region, the EZ region, was negatively charged, and the region beyond was positively charged. So sort of acting like a battery. And um, and the way this happens, we were able to deduce is, it all starts from water. This is a water molecule, and the water molecules are all over the place. And what happens is that when a certain energy comes in, and I'll describe that energy in a moment, it breaks the water molecule into two parts. And one part is the oxygen is sitting here with a valence of -2, and the hydrogens are +1. So if you break it here, you get -2 +1, a net of -1 charge, and this is +1. And what happens is that this fraction of the water molecule, these fractions gather together in this region to build this negatively charged EZ. And these positive hydrogens get cast into the water beyond, or pushed into the water beyond. So you've got the negative charges separated from the positive charges. And these are essentially like terminals of a battery, with negative here and positive here. So you've got energy that comes from this. And the way it builds, what we, again, were able to deduce is you have a material and you start with water. And out of this water form these EZ layers, that is, remember those OH minuses, build together to form the first layer. And after the first layer forms, that layer acts as a template for the formation of the second layer and the third and so on. And these hexagonal layers keep building from the water one at a time, and they keep building and building and building, as you see here. And if you were to take a look at one of these planes, it would look something like this. This is the honeycomb, the hexagonal features that you see all over nature. So this is not really a surprise, with the hydrogens here and the oxygens here. And so it turns out that the structure is no longer H2O. It can't be H2O because H2O is neutral. And we know that this is negatively charged. And therefore, it turns out if you count the number of hydrogens and oxygens in the unit cell, it's H3O2. So this is these EZ layers, or fourth phase, is actually H3O2, not H2O. And if you think about what's inside the cell, which I'll get back to later, inside the cells, they're crowded. I mean, it is crowded inside the cell. This is a gross underestimate, this cartoon, of what it's like inside the cell. There's practically no room for water. All water molecules are near one protein surface or another. Therefore, they're going to be this negatively charged EZ water practically inside the cell. And I'll get back to it in a moment, but I want to digress just for a moment to talk about something that is of great interest to us, and maybe of some interest to you, the idea that EZ water could conceivably store information. So if you think about what's required, what do you need to store information? First thing that comes to mind is perhaps a thumb drive. What's inside a thumb drive? Well, it's a planar structure consisting of silicon-based transistors. And these transistors have two different states, on state or off state, so, or 0, 1. So the critical feature of this kind of memory is an ordered state where each entity in the ordered state can have, in the ordered array, can have two possible states. Now think about EZ water and whether it corresponds to that. So we have the so-called nucleator, which is the material with a hydrophilic surface, and here's the water. And remember, we have these layers, and these layers are very well organized, just like a crystal, in fact, a liquid crystal. And they consist of oxygens and hydrogens. And so the first criterion is met very well. This is an ordered structure. And in fact, the ordered structure is a three-dimensional rather than a two-dimensional structure, very closely packed. And the question is, well, do either the oxygens or the hydrogens have two states? And the answer is absolutely. The oxygens have not only two states, but any chemistry textbook will tell you that each oxygen atom has 5 possible oxidation states. -2 is the usual. There's -1, 0, +1, +2. So in terms of information stored, potential information storage density, this is amazing because first of all, it has 5 different states instead of 2 different states. And second of all, it squeezes so many stacks of layers into a confined space. And we computed that if this were to act as information storage, theoretically at least, we don't know if practically, the storage density exceeds the storage density, current storage density by a factor of 9 orders of magnitude. That is 1 billion times the storage density. And so it's no surprise if one day perhaps computer memories will consist of easy water. Now, in terms of memory, a lot of people know about the work of Masaru Emoto from Japan, who passed about 5 or 6 years ago. And he demonstrated that if you put intention or various kinds of energy to water, that it influences the water in such a way that the ice crystal's shape, if you freeze the water, differs. Now, Emoto was no scientist, and he was a spiritualist. He never claimed to be a scientist, and a lot of scientists don't take his work seriously at all because he would freeze, say, 50 petri dishes under one condition or state, and he'd pick out the one that of the 50 that best illustrated what he wanted to show. And this kind of approach, of course, is not a scientific approach. On the other hand, there have been some meta studies that looked at what they found and appear to confirm that there's something interesting going on there. And I've been following their stuff closely because the EZ actually has some capacity to store information, as I suggested. And so there is a possible theoretical reason why Emoto and people might have found what they found. So what did they find? Well, just a couple of examples here. You play a Mozart symphony to water, you freeze the water, and you look at the ice crystals, and you see something nice that looks like this. John Lennon is perhaps even imagined, is even nicer. And this is thinking about peace. Put your attention to the water, and as you put your attention, you're thinking about peace in the world. And again, a beautiful crystal. Thank you is especially beautiful. You fool produces something rather ugly that is actually similar to Tokyo water in its quality. And here's some heavy metal music. So this is typical of the work of Emoto. And as I said to you, there are a lot of people around a lot of scientists around who don't really take this work very, very seriously. However, this field has attracted some heavyweights, and a couple of the people who have been involved in the idea of water memory have been a couple of Nobel laureates, Brian Josephson in physics, and more recently, Luc Montagnier, who won the Nobel Prize for discovering HIV. And this is Luc here. And he's been working on studying memory of water, particularly as it relates to human beings now for pretty much for a decade. And his results are nothing short of astonishing. And let me just illustrate here because it's worth a moment because of what he's found. And so what he does, he has 2 containers, and these are closed containers, actually more securely closed than has been illustrated here. So there's no possible crosstalk or chemical contamination between one and the other. They're factory sealed, very well sealed. Here, he's got sample DNA, a short sequence of DNA, 100 base pairs, perhaps, of a particular DNA. And often the experiments are done where the DNA is pretty highly diluted, similar to what you do in homeopathic preparation. It doesn't have to be, but he's gone to very, very high dilutions where pretty much all that's left is water here, and at least statistically, no DNA. And his argument is that the information is being transferred somehow from this container to this container, not through chemistry, but some sort of energy. And that's illustrated here. So A is sitting next to B, and he imposes some kind of generic energy, electromagnetic energy, 60 hertz, 50 hertz in Europe, 7.8 hertz, whatever. And he does that for 24 hours. And he says, he hypothesizes that the information from this DNA, or from the water surrounding the DNA, somehow is getting transferred, some information exchange that is transferred to container B. So then he throws away container A, takes container B, just water, pure water, with supposedly with information, information about this DNA. And he puts it into a container with the DNA precursor material, which then gives you DNA, and voila. And the new DNA matches, the sequence of the new DNA matches the sequence of the DNA in A. In other words, somehow the information from the DNA or the water around the DNA has gotten transferred to the water. So as you can imagine, this was not well received by mainstream scientists. On the other hand, it's been confirmed by at least 2 other groups. And so it appears that there's something going on there that is very interesting, that indeed has to do with the memory of water. And this is done by Nobel laureates, so we do pay some attention. Now, a point of relevance of all this that I just want to tell you about, it. Again, this is a digression on a digression, is that it's possible, it turns out, to take easy water. Remember, this is water. This is a liquid crystalline water. It's possible to take it and solidify it at room temperature. So what am I saying here? It's possible to take a kind of water and make a solid water, that water at room temperature, sort of like ice at room temperature. It's not ice, but it can be solidified. And that was done by Vittorio Elia from Naples, who published a few papers on the subject. And of course, we were tempted to see if we can reproduce it. But what he found is just astonishing. If you collect the easy water, of course, it will always be mixed with some ordinary water. You just put it into a container and let it sit over time. The solid EZs coalesce over time, forming cloud-like structures. And then if you go through the process of dehydration, of getting rid of the water, then this becomes a true solid. Interesting that it looks like clouds. And I think it's more than just a coincidence. That's the case. So this is what he finds. This is solid, easy water at room temperature. You can take it, pick it up with your hands. It feels like kind of fine powder. This is again from his lab, and we've undertaken experiments in our own laboratory, in my lab at the University of Washington, and we could reproduce what he's done using slightly different technique, and also adding something to it. What we found out using mass spectrometry is that this cannot be caused by some contaminants. We checked 3 different methods of producing this, and we checked for contaminants, and in no case were there contaminants in any kind of concentration that could cause this. So it's real. And so when you go back to the idea of maybe having a computer memory stick like this, it could be filled with easy water, but it could also be filled with solid easy water, which is perhaps more practical in the long run. So a prediction is that this easy water has tremendous capacity to store information. And because it fills your body, this water, your body also has capacity to store information. And so I think this is the future. So remember, it's not just stuff that's filled with easy water, but healthy people like these, totally filled with easy water. Every cell in their body is filled with it. So going back to the question from which I digressed is this easy water physically distinct from bulk water? Remember, I presented a long list, but no time to go through all of it. But the answer is yes. Anybody can check the references and see. It's a layered honeycomb structure as best as we can surmise with information storage capability. And this information, I think, is so interesting and is grist for the meal of the future. So I mentioned to you earlier that the EZ is like a battery, plus and minus at one side and plus at the end. And you know, everybody knows that batteries need charging. And so what is it that charges the water battery? It took a while for us to find out, I must admit. There was a student who found out, and it turns out it's light. And the student experiment looked like this. This is real data, and this is obviously a cartoon, but he took some hydrophilic material and he used Nafion, which is a polymer, a little bit similar to Teflon, but has charge groups to it. And he was looking under the microscope and he could see the exclusion zone here and the microspheres that were excluded right here. And generally, the boundary between the exclusion zone and the region beyond is a straight boundary parallel to this nucleating surface right here. So it would the boundary would be a straight line parallel to the surface. And it was, and is. However, he took a lamp that was sitting right next to him on the bench and he shined it on the preparation. And he called me in to show me what he saw. And it was amazing because, as you can see, the region that was illuminated, the exclusion zone grew. And, you know, very substantially with, in fact, a really, really weak light. And I, you know, I was dumbfounded. I couldn't believe that this student had found the answer to the question that we couldn't answer for a while. It's as simple as it's light. I asked him to take the light away. He took the light away, and this extended region went right back to the original, and the EZ was then boundary was then straight as it ordinarily is. So we were inspired to figure out, well, which wavelengths of light are the most critical. And we found that we studied wavelengths ranging from the ultraviolet through the visible spectrum way to longer wavelengths of infrared. And we found that by very far, the most effective was infrared. Now, so infrared, it's light, very much the same as visible light. It's just it's a longer wavelength, and our eyes can't detect it. Otherwise, it's pretty much the same. And it turns out that it's all over the place. Most of us don't think about it, but if we were to turn off all the lights in the room, so neither you nor your cell phone camera can see anything, and you were to whip out an infrared camera, you could see a beautiful image of everything that's sitting in your room, including yourself, if you happen to be sitting there, because everything is generating infrared light. It's always there. It's essentially, practically speaking, it's impossible to get rid of it. It's always there. And this is why it's used by the military as a night light or as a night detector, because it can basically see in the dark. It just picks up the infrared that's always there. And because and the infrared we found is something like almost 1,000 times more effective than the visible wavelengths in terms of its capacity to build easy water. So it's always there. And since it's always there, it means whenever you have a hydrophilic material that's sitting next to water, you'll always have some easy water. And if you add more infrared light, you get more easy water. And if you were to remove it, it would go back to the default situation. So, so you've always got easy water. So the energy for buildup of easy water comes from light, photonic energy. That's what's responsible. Photons, photon energy is responsible for easy buildup. And this energy orders the water. You need energy to go from chaos to order. It orders the water and it charges the water battery. It separates the charges. So therein lies the energy. So it's actually rather simple. You know, you just lie on the beach, wherever that beach happens to be. You know, it could be New England today, or it could be Hawaii. You just lie there and you get filled with energy. The battery gets charged. Now, so if you're still with me and you're thinking, if the battery is getting charged just by lying there, basically in the sun. Remember, the sun, half of the sun's energy is infrared energy. When you feel heat, the heat is essentially, not exactly, but essentially the same as infrared. Half of it, 50%. So you're getting quite a lot of input. And so the question is, if this is filling with energy, you'd expect that this battery, if you will, ought to be able to release the energy somehow and do work. And so a question arises, can this energy that you get just by exposing yourself to infrared, can it be harvested in some way? And I think you've probably never seen a glass of water doing work. And so you think, wow, no, I'm going to show you how a glass of water can actually do work. Let me show you. Maybe not a glass, but essentially a container of water. So here's an experiment. This is done by another undergraduate, and it was Arthur Yu some years ago, freshman, sophomore, working in the laboratory as a student volunteer, and we had just learned that Nafion, which we like to use a lot because it's easy to use, it came not only in sheets but also in tubes. And we wondered, it has curvature, and we wondered, despite the curvature, is it possible that you could still get an EZ that builds outward from the surface and inward from the surface? Within a week, Arthur had the right answer, and the answer was yes. Indeed, he could see an exclusion zone outside and inside, despite the fact that it was not flat, that it was a cylinder. So he was happy about that, and before we got together to discuss what to do next, because he had already done what I had suggested him to do. He was playing around in the lab, and one day, a week or two later, I was sitting with an important visitor. I think the visitor was important, but maybe not the most interesting visitor. And we were chatting in my office, and the door was closed. I don't usually close my door, but I think I remember for some reason it was closed or almost closed. And Arthur Yu came running in, and usually the students would kind of knock at the door politely, "Are you busy?" or whatever. But he just threw open the door and said to me, and I was having my discussion with this visitor and such, and he said, "I found something interesting, and I wanted to tell you right away." "Okay, Arthur, tell me what's so interesting." So he said, "I'm watching this tube in the microscope, and I can watch it. I put little particles in the water," he said, "and I can, with a microscope lens here, looking upward, I can see what's going on. And I noticed that the water keeps flowing through the tube," he said, "and it keeps going. It doesn't want to stop." He said, "I thought this was kind of interesting." And I thought, oh my God, if Arthur is right about this never-ending flow, and we confirmed that indeed he was right. Afterward, uh, this is amazing because usually if you try to drive flow through a tube, you need energy. The water has viscosity, and you got to kind of push it through. It's the same way that the heart is pushing blood through the large arteries, and it takes energy to do that. And so where could the energy be coming from? Well, there's no pressure differential here because the height here and the height here are the same. So there's no pressure head that is larger here than here to push it through. So the only thing that I could think of was that this was good evidence that that we were right, that indeed that the energy that's coming in, the energy from the sun or from any kind of infrared source, is being absorbed by the water and converted into some kind of mechanical energy, because where else might energy come from? So we were excited about this. And the way the experiment is done, it looks like this. You use water, make sure there are no air bubbles in it. And after it's full, you stick it into a container of water plus these microspheres and look in the microscope, green light to reduce the total amount of light. And what you see is something that looks like this. The Nafion tube is here. Here's the EZ, same on this side. And all the microspheres are here, and the microspheres keep going through. We've had it going for a day and a half without stopping. The video stopped. A day and a half. And then someone said to us, we keep our ears open, someone said, oh, I bet this is some kind of special feature of Nafion, the polymer Nafion, which seems to have some unusual properties. You should really try some other kind of material and see if it's general or not. So we searched for other kinds of tubes of the same size, hydrophilic nature. We found difficulty finding something that was useful, and so we made our own. And the way to make it, again, a clever student did this. You take a gel. Before it's gelled, as a liquid form, you stick a wire in. It's a little chunk of gel with a wire. And as it's gelling, you pull the wire out. And you wind up with a little chunk of gel that has a tunnel running right through it from one end to the other. And so basically that tunnel is just like the tunnel here, and it's just got a lot more stuff on the outside than just a thin layer of, say, Nafion. So, and we would take this chunk of gel with tunnel and then stick it in the water with the microspheres. So it looks something like this. So this is a gel, polyacrylic acid gel here and here, and here's the tunnel. And you take this chunk of gel with tunnel and you stick it into a larger chamber containing water and microspheres. And before long, you see an EZ that's built up here, an EZ that's built up here, and all the microspheres get pushed to the center like this. And if you look at the video, what happens is this same result as we saw with Nafion. In fact, we've now seen the same result in something like, I can't recall, 8 different gels by now. So then someone said, well, if you think that this is driven by infrared light, why don't you just increase the light and see if it goes faster? And so we did that experiment and we published the paper 4 years ago. I think it was, we could achieve a 5 times increase of speed just by turning up the light. So we're confident of this, and the mechanism, the way it works is that we deduced is that here's the tube, and here are microspheres, and here's an exclusion zone, EZ here, and EZ there, and this EZ we know is negatively charged, and the bulk waters here is positively charged, and because of all these free positive charges, they're free to move. The negative ones are stuck to the surface. These are free to move, and it builds up inside the cylinder, and it wants to escape. They want to repel each other. So the escape will either be this way or this way, wherever the concentration is highest. And that's what initiates the flow. And then as the water moves out this way, more water gets sucked in this way, and the process perpetuates. So we have a situation like this. Work is done in order to push the fluid through the water. Energy is required, and therefore energy absorption is necessary. So water is really a transducer of light energy. Now, do we use radiant energy? If you were Mother Nature, and by we, I'm talking about humans, but I'm also talking about vegetation and plants at the same time. But in this case, if you were Mother Nature, you were very successful with plants and even unicellular organisms, all of which use light as energy. Light is the energy. And you decided one day, I'm going to invent animals, and animals can forage. They can get their energy from food. Would you continue to use light as a source of radiant energy, or would you just throw it away? I think being conservative, thoughtful, and intelligent, that Mother Nature would keep this light mechanism in some kind of reserve, either using it together with the food equally, or keeping it as a background supplier of energy, or what. That would be the conservative way to go. So now, when you think of us, most of us look better than this guy, but we're receiving infrared energy all the time. And so where might we capitalize on this energy? And one thought was, well, maybe in the cardiovascular system, because as you know, the capillaries are pretty close to the surface. And so it's possible somehow, maybe, that this energy could be used to help assist the heart in the cardiovascular system. And at first, I had mixed feelings on this, because as a graduate student, I studied the mechanics of the circulation. And I thought I had it all worked out. And then I went to I went to Russia to meet my friend Vladimir Vyakov at Moscow University, and he quickly introduced me to the guy from the neighboring laboratory. And the guy was telling me a story about some problem in the cardiovascular system. I must admit, I had my nose up in the air, because I was thinking, I know what there is to know pretty much about the mechanics of the cardiovascular system. And this guy is telling me there's a big problem. So, okay, tell me what the big problem is. So he starts telling me, and he had me convinced in 5 minutes that he was right and I was wrong. The problem is this. The problem is that the red blood cells that need to pass through are 6 or 7 micrometers in diameter. But the capillaries are smaller. Typical capillary diameter in a healthy young adult goes down to 3 or 4 micrometers. So, how is it possible that you could take this giant galumphing red blood cell and push it through the capillaries? Well, they calculated how much energy was required, and or how much force was required. And they concluded that if the heart is responsible for doing this, the heart would need to develop a pressure of something like 1 million times the pressure it ordinarily develops. And you might, parenthetically, that's high blood pressure. So there has to be something else. And he was arguing there's got to be something else, not just the heart. There's got to be something else that pushes the blood along. And at first, I was a bit skeptical still. And then my student was working on the project, began reminding me. Let me go to the, whoops, no. Reminding me that he could find papers, something like a half dozen papers, showing that, I'm sorry, that if you stop the heart, what you expect is the blood flow will stop, but in fact, it doesn't stop. It keeps flowing at much lower velocity, but it keeps flowing, and therefore there has to be something beyond the heart that's doing the propelling. You see, and this video just illustrates the seriousness of the situation. So here's what a red blood cell is supposed to look like, and here you see other red blood cells. This is muscle tissue, and you can see the striations here, and here is a a capillary and another capillary and another capillary, you can see that the red blood cells are squeezed down. And so in order to get through, they need to squeeze. This guy's having a really hard time of it. And therefore, what they concluded is you need energy to take care of. There's something beyond just the heart, because this happens even if the heart is stopped. A half dozen different papers. So I'm thinking after this that my goodness, in the laboratory, we learned just recently that radiant energy can drive flow. It builds EZ, and the EZ is responsible for creating the flow. And the question is, might the same radiant energy help drive blood flow? Skip this one. So we did our experiments, and we did it with chick embryo. And this is a 3-day-old chick embryo. 3-day-old meaning that cardiovascular system is fairly well developed, but the regulatory systems, chemoregulation, neuroregulation, is not well developed. And so you can lop off the top of the eggshell, and he developed, my student Li Zheng developed a microscopic system to be able to visualize these vessels. And so what he did was, step one is to stop the heart. And you can do that just by injecting some potassium chloride. And so here is the blood flow as a function of time. And if you block the heart, the flow diminishes this way. And it diminishes, but it doesn't go to zero. It diminishes to some lower level. And then to test to see whether our mechanism applied, the idea was to test its signature feature. And that is, if you apply infrared, it should speed up. And this is exactly what we found, a factor of 3 or so speed up. And then when you remove the infrared, it goes back down to baseline. So we think on this basis that what's going on is inside your body and my body. The major driver of the circulation is the heart, but it's not the only driver. The secondary driver are the blood vessels themselves, which can drive flow simply based on this flow mechanism that we discovered in our laboratory that's run on infrared energy. And obviously this has, important implications for your health and for my health. So we think that radiant energy does help drive blood flow, and probably similar or almost certainly similar for plant flow, because remember, we have the EZ that's working here just as it works in the inside of the Nafion tubes and all the gel tubes that we studied. It's just, this is in the vertical direction, and we did confirm in the laboratory that this mechanism that we uncovered works just as well in the horizontal or vertical direction. So we think this explains not only the flow, but also the seasonal dependence of flow, because, in the springtime when it gets warm, you've got plenty of infrared energy. Infrared is essentially equivalent to heat or warmth. And this is what drives the flow. And so in the springtime, when you want the flow to happen, the flow begins to occur because the energy source is available, and that keeps the leaves green. In the autumn, as it begins to cool and the amount of infrared diminishes, the EZ diminishes because it doesn't have the energy to keep it going, and the water flow in the xylem stops and you get dehydration. So I'm not suggesting from all of this that we as humans, for example, do what the plants do and photosynthesize. But the first step of photosynthesis, is the splitting of water into positive and negative. And this may indeed be what's going on, not only in the plants, but also in us. So more generally, EZ is really I've been trying to allude to the fact that the EZ presence is central to so much of what goes on. But how does it facilitate function exactly in us and you might say in plants? So in green algae, we know that there's photosynthesis that goes on. The same is true in some bacteria. And of course, green plants, the rainfall or irrigation water gets absorbed by the roots. It gets driven into the leaves of the plants. It's then subject to the infrared energy, and the infrared energy splits water. Step number one, 100% efficient. And it seems the same process is going on beyond just plants. It seems that the process that I've described to you is very similar to this, almost the same. And so you've got a material next to water, and it's separating into the negative and positive, and this being the exclusion zone or fourth phase or easy water with protons here. And so there's a lot of potential energy that you see here that can get released inside the cell. And so you have a cell, and inside the cell, you've got, as I tried to describe before, you've got solids, most of which are proteins, about 70%. And these proteins pretty much are packed inside the cell. But next to each of these is water. The water should be EZ water because the layers are very close to the protein surface. And because it's very crowded, essentially all the water in the cell is negatively charged EZ. And that's why, I think, if you stick an electrode in the cell, any cell, you get negative electrical potential. If you read the textbook, you'll find it has something to do with the membrane, channels, and pumps. And I don't have time to argue here that that is not correct, but we have a paper that discusses that. And so the potential energy here is kind of, as I described here, these negative charges all want to get away from one another. They repel. And so because of that, you have potential energy. And the potential energy is involved in protein folding. The protein folding is what's responsible for what the cell does. In a muscle contraction, for example, the proteins will fold, thereby giving rise to a contraction. And so it works something like this, the folding or unfolding. You have a protein, and next to the protein is the EZ water shown here. And so when let's say muscle cell, it could be any cell, example, muscle cell is stimulated to contract, the EZ water melts into ordinary water and the protein folds. So together, it's the melting of the water, the transition of water into ordinary, from EZ water to ordinary water, that allows all this to happen. And then when the contraction is over, or whatever the cell does is finished, it returns to its ordinary state with EZ water buildup. And that's where the energy comes into play. You need energy in order to get it back. And so in the case of muscle, if you don't have enough energy, the muscle will remain contracted. You'll have a knot in your muscle. If you go to the right side, if it happens that your cell doesn't have EZ, then it can't return. It's basically impotent and your cell is not functioning. So you need EZ water to fill your cell. So the potential energy from EZ is really what drives, at least in part, the work of the cell. And just to go through the sequence again, light builds the EZ. EZ contains negative charge. This negative charge gives you potential energy because it all wants to escape. And the energy is responsible for the work of the cell or the folding of the proteins. So basically, light is responsible, in part at least, for the work of the cell. And so if you ask, where do we get our energy? We get our energy, of course, from food, but we also get our energy from light, which is absorbed by the water, and which builds EZ and gives us energy. Well, should this matter to you? Well, I would think so. Light matters, is what we've learned from this, and water matters even more than you come to appreciate. And so what you might want to know is, in order to preserve long-term health, what builds EZ water in your cells? And I'm just going to run through these because they're actually pretty simple. Some are interesting. The first is just water, drinking a lot of water. It's the raw material for the body to build easy water. So you need to drink enough of it. Green juicing. So you take the plant, take the leaves, squeeze the leaves to squeeze out the cell water, drink it. It may taste awful, so putting in some strawberries or something to make it palatable really helps. But what are you drinking? Well, you're drinking the water from inside the plant cells, fresh, robust plant cells containing lots of easy water. And therefore, you're drinking directly the water that your cells need. And so you're replenishing your cells with easy, and your cells should be functioning very well. And I've heard from many health practitioners, this is the single thing that you can do that's easiest for preserving long-term health. There are also various substances that are known to improve health, dating back from Ayurvedic times. And turmeric is one of them, coconut water, and several others. We tested the idea that the way these actually work is that maybe they're so effective, so universally effective in so many areas of the body is because they impact something that's all over the body, and that is water. And so we wondered whether if you add a little bit of turmeric, you actually get an expansion of the EZ. And we found exactly that and published it. It's not just turmeric, we also found the same coconut water, basil, ghee also is extremely effective in building EZ. So yes, it seems that if you eat the right foods, it should build EZ in your body, which should sustain good health. Your cells should function properly. In Seattle, where I live, we don't see the sun too much in the wintertime. When the sun comes out, people are smiling. And the thought is, well, you know, it's a psychological effect, and it might well be, but it also may be that since the sun is emitting 50% of its energy in the infrared, and this is exactly what builds EZ in our body, exposing ourselves, including our brains, our head to sunshine, builds EZ in our brain. And we feel good, we feel better, because that's the default state of our brain. Could be as simple as that. And going to more extreme, a sauna, as the Finns pronounce it, was a sauna. It's hot. The heat is producing infrared. We go in there, take off our clothing, expose our body to infrared, and we may start with a headache and muscle pains and et cetera, and we walk out feeling like $1 million. A possible reason is we're exposing ourselves to infrared. Infrared builds EZ in our cells, and our cells are suddenly functioning better, back to normal. And finally, grounding, or so-called earthing. What we're doing is we're connecting ourselves to the negative charge of the earth. Earth has a net negative charge. I was surprised to find it, but the evidence is absolutely overwhelming. It's just that we don't follow it, don't pay attention to it. So by connecting ourselves electrically to the earth, like by walking barefoot on the beach or taking a mud bath, or just connecting ourselves electrically to the earth through various means, basically the negative charge of the earth is free to run into our body, building EZ. And therefore, many people have reported this as a powerful expedient toward restoring good health. So these are some of the things that you can do to improve your health. I end with practical applications, getting energy from water and sunlight. So we have a situation like this. We have a hydrophilic surface, water. We have EZ, bulk water. You've seen this before. Put 2 electrodes in, should be able to give you electrical energy. And we've demonstrated in the laboratory that it can. So here you have little dozen or so cells of water with Nafion. And each cell contains 2 electrodes, one in the negative, one in the positive region. And here's a switch. Here's a magnifier, and behind the magnifier is an LED. And so you turn the switch and the LED lights. And this is energy from water and light, completely renewable and exciting. And a long way to go between laboratory demonstration that you see here and practical implementation. But we're working on it. Second one, or I should say second and third, getting drinking water from contaminated water. So here is a schematic of the device. You have inflow. This is now water from with contamination with, choose what you like, pharmaceuticals, microplastics, whatever. And we use microspheres as representative contaminants. So what happens is you have a tube, like a Nafion tube or some other material. We use other material, which is cheaper. And all the junk gets excluded from this region, and this region gets shifted to the center. And we then use a so-called differential extractor. It's nothing more than two concentric tubes to do the separation. So this collects the junk, and you can see the representative junk here. As I said, we use microspheres in our tests. And what you collect here is junk-free water. And as you can see here, this is one representative example. This has got a lot of junk, this has little. We've been able, in the best situation, to get a separation in, of 200 to 1 concentration in one single pass. And we've spent quite a bit of time and made progress in developing it. We're not yet there. But the exciting part is the possibility of separating salt from water. And so you can imagine taking ocean water and putting in one of these tubes and getting out water that has no salt, separated from salt. Theoretically, it should be possible. We've made some progress, but not as much as we hope. So imagine the situation. All of this separation happens not from a real filter of some sort, no physical filter. We call it filterless filter, but it happens from the energy of the sun. So you can, in theory, you can separate salt from ocean water, in theory, at least, in principle, based not on energy from oil, which is done a lot, say in the Middle East and other places using in the reverse osmosis process, but actually from the energy from the sun. And where you need this drinking water most, there's lots of sunshine. So we're excited about that as well. So I conclude by saying that we found that although we've learned that there are 3 phases of water, we found there's a 4th phase. I've only had time to go over a bit of the information. And I put this 4th phase here in between ice and the water phase because we've learned something. We learned, first of all, that the structure of this is not too different from this. Easy and ice share, they're both hexagonal. They're not the same, but they share some features. And we found through experimentation, a surprise, that if you want to freeze water, you can't take water and go directly to ice. You must pass through this easy phase. And if you're to melt ice, you can't go from ice to water. You must go through this easy phase, this transitional phase. And that's why it's put between these two. And this has, all of this has broad implications. The central feature, I think, of all of what I've been talking about is that when you have water, any kind of water, whether the water is in you or in a plant or in a tree, the radiant energy is being absorbed by the water and used by the water. And there are biological uses. I talked about blood flow, for example, using that energy to help drive blood flow. It's also true in, or maybe true in pretty much all aspects of chemistry, because the chemistry book says nothing at all about the role of light, infrared energy, charge separation, ordering, et cetera, which means that if what I've suggested to you is correct, or even partly correct, a lot of the interpretations of what you read in the chemistry book may need revision. In terms of weather, you know, we never know, at least in the Seattle area, we often hear a 50% chance of rain tomorrow. Well, it's not very helpful. It doesn't tell us whether to bring an umbrella or to leave the umbrella home when we go to work. We'd like to know better. And I think part of the reason for this is that the forecasters or atmospheric scientists take into account two variables mostly: temperature, pressure. Never, I guess I can say never, have I seen a discussion of the charges that exist in clouds at all as being at all relevant. Yet we know from lightning and thunder that the clouds contain plenty of charge, and even plain old lovely cumulus clouds that we see up there are just full of charge. Charge is so tied to clouds that if you fail to take account of that, I think you're missing the big part of the picture. And so in the future, and a forthcoming book of mine will be discussing this, the importance of charge in weather. In health, I mentioned to you that your cells are full of easy water, and they need to be filled with easy water all the time in order to function properly, in order to function in terms of health. And in terms of food, if you want to prepare food, either store it by freezing it or store it through dehydration, it's nice to understand how these these features of nature actually work. And then from a practical point of view, I talked about filtration that comes out of these scientific studies, including desalination, exciting, and getting electricity from water, from sunlight and water. So we're really excited about this. And before the final slide, I'll just mention, as I'd like to do, our Institute for Venture Science. And it funds promising ideas that may bring scientific revolutions. It funds promising ideas that challenge long-held views in science that may have outlived their usefulness. And so we're excited about it. This is the URL. It's very simple: IV, as in intravenous, ivscience.org. And we've identified from 200 pre-proposals, very thoroughly vetted, 5 projects, 5 exciting projects. And we're looking for donors who would like to, who have done well and would like to share, give back in to a cause that will undoubtedly help solve the many problems of that the world faces today. And I end with this slide. I've talked about perhaps 5% of what's in the book. There's a lot there about water. I find it all very exciting, and I think I will end here, leaving 5 minutes or so for some questions. Thank you very much.
Well, thank you very much indeed. That was very engaging. You gave us a lot to think about. The first question is, do you have some other resources that you would recommend for follow-up reading?
Yes, I do. Yeah, this is the first one I'd recommend. And then the book, the 2001 book, Cells, Gels, and the Engine of Life, focuses more on, it's earlier. We hadn't known about some characteristics of the 4th phase, where it talks about water structure and how critical that structure is in biology. So if you have some interest in biology, have a look at that. On the web are many presentations over the years. I don't keep track of them, but there are many there. And some of those share different features of what's in the book. So I recommend that. I also recommend our website. It's called Pollack. If you just search under pollacklab.org, you'll find it. And of course, those who are like seriously interested in the science, please read our many publications that exist throughout the literature. So that's where I would go if you have interest. But I would certainly start with this book.
Okay. So I think we answered the question about what determines the direction of flow through the Nafion tube. But could you explain a little bit more? You thought it was perhaps an imbalance or?
Yeah, I actually didn't really explain it in detail. It's a really good question because everything looks symmetrical, and so why should the flow go in one direction versus the other? And I think the answer is that it's not entirely symmetrical. If you do the experiment the way we do the experiment, we take a long run of Nafion tube, for example, and we'll cut a segment, maybe a few centimeters or sometimes as short as a few millimeters. And when you make this cut at either end, if you look in the microscope, you'll see that it's not it doesn't turn out to be a right circular cylinder. The cut at each end is frayed. It's no longer, the cross section is no longer a circle, it's more an oval. And one side always differs from the other side. So you have some asymmetry. And then further, you have asymmetry because the light that's coming in, especially the infrared light, will inevitably emerge more from one side compared to the other side. For example, you may be near a window or may be near a heater on one side, and therefore, almost certainly when you do the experiment, you'll have asymmetry. And there's asymmetry in the cardiovascular system too. And so as long as you have asymmetry, something will be different from one side versus the other. And that asymmetry should be enough to drive the flow in one particular direction. And once it starts going, the water is pushed out, let's say the right end, and then it gets sucked in the left end to replace what's been pulled out. And so the process perpetuates itself. So yeah, at first we scratched our head to figure out the reason why it actually works. And I think the reason lies in asymmetry. I will say that in perhaps 10 or 15% of cases, we get no flow. And in those cases, I think that we don't have enough asymmetries to start the process going. So that's a more extended answer to that question.
Interesting. Yeah. I was wondering if you had reversed the Nafion tubes or switched it 180 degrees, would it continue at the same flow in the direction it had originally?
Yeah, well, yeah, it would flow in the same direction relative to the tube that it had originally. Yeah, so not the same. Hard to describe this. Yeah, with reference to the tube, it remains the same.
Right.
So if you got a fire hose and you redirect the fire hose, it'll come out the way you like it directed.
Excellent. We're coming up right on an hour and a half. So with respect for everyone's time, I'll wrap it up here. Again, thank you so much for all that great insight and information.
Well, thank you, Doug. I appreciate the opportunity. And yeah, thank you for the chance to send a message. I think there's lots of application of this message to plants, agriculture, soil too. Thank you.
Yeah. If there's any more questions, would you like to share your contact information?
Sure. Email. Well, I'm really easy to find on the web. Nothing is hidden, but it's ghp@uw.edu.
Excellent. Thank you. And we'll include that link in the description as well.
Sure. That's fine. Happy with that. So yeah.
Announcements before we go. We have an evaluation. We would really love your feedback. We read every single one, so please help us make our programs better. And additionally, it's a good time to check out our vendor marketplace, and we encourage you to visit with them and learn more about their products and thank them for being here. If you haven't already checked out the auction, the online auction, that is still going all through today and this week. So you have time to see what's available and go ahead and see if there's anything you would like to bid on. We have the rest of the afternoon, a series of events and throughout the week. So please come back and be sure to refresh your program booklet to make sure you have all of those up-to-date links so you can catch the next session. And that's it for us. Again, thank you so much for being here, and we look forward to seeing you next time.